Jens Wiltfang is a graduate. Wiltfang. Jens Wiltfang is a graduate from the Faculty of Medicine of Hanover, where he obtained a PhD in psychiatry. He is the director of the Department of Psychiatry and Psychotherapy at the University Medical Center in Göttingen. He's a neurologist and psychiatrist with wide experience in the field of neurodegenerative diseases. His activity includes the clinical characterization of patients with dementia and research on the biomarker discovery and validation. In particular, his main expertise is on the field of cerebrospinal fluid and blood biomarkers for early diagnosis of neurodegenerative diseases, with special interest on Alzheimer's disease and Parkinson's disease. So, I'm very interested in that. So.
Yeah, thank you for the kind invitation. So I'll start right away. So the topic of my talk is predictive molecular diagnosis as a prerequisite to set up novel clinical models for preventive treatment. And why is that? So current needs are treatment of Alzheimer's disease starts much too late, and most importantly, there's strong evidence that we miss critical time windows. We definitely need novel preventive treatments. What we have available nowadays is purely symptomatic. We need to identify novel subphenotypes of multigenetic sporadic AD. They are genetic cases, but they make up only 1% of also cases. So the predominant cases are multigenetic sporadic cases, which may then respond differentially to treatment interventions. And in this area, biomarkers are very promising. And last but not least, we need to identify novel treatment targets. And I will give an example in my talk that research primarily dedicated to diagnostics may come up with novel therapeutic targets as well. Now, this is the amyloid cascade hypothesis of Alzheimer's dementia. Our group does not claim that this is the etiology. For multigenetics, this is still open for debate, but at least those biomarkers are highly correlated surrogate markers, and thus they can be used for diagnostic terms. So here you have the amyloid precursor protein with a large extracellular domain, which then is cleaved by beta and gamma secretase to give rise endoproteolytically to a beta peptides. Two species are depicted here. Actually, this is much more complex, as I will deline later. Beta peptides are prone to form soluble oligomers via amyloidogenic intermediates. We will tackle this by a new assay, which I will present at the end of my talk. So this is molecular neuroimaging. And then soluble oligomers precipitate as insoluble amyloid fibrils, which finally form the beta amyloid plaques described by Alzheimer. And correlated to this precipitation is a selective decrease of 1-42 in cerebrospinal fluid, because 1-42 makes up the majority of those fibrils and plaques. So that's an important take-home message. We have a selective decrease of 1-40 because 1-40 is more or less unchanged. So the ratio 1-40 to 40 is declining in CSF. Then in the vicinity of the plaques, we have a strong neuroinflammation, which is a promising novel therapeutic target. And within the neurons, we have the formation of neurofibrillary tangles, which have also been described by Alzheimer by staining techniques. Of course, he didn't know the molecular constituents. And with emerging synaptic and later neuronal degeneration, we have a release of the molecular constituents of neurofibrillary tangles. This is tau protein, most importantly, hyperphosphorylated tau protein species. So second take-home message, in CSF, tau goes up, more specifically, phosphotau. Total tau is a more unspecific measure of neuronal destruction. Then those patients who are on their way to dementia, unfortunately, they have to pass a preclinical state of mild cognitive impairment where cognitive deficits are evident, but they are not so strong that they do not deteriorate everyday life performance. Now, this is a famous landmark paper of, this is supposed to be Hansen in Lancet Neurology from the group of Kai Bleno in Sweden, who they conducted a multi-center study where patients at several clinical sites presented in the prodromal state of MCI. Then the CSF dementia biomarkers were estimated at baseline 1 to 40 total tau phospho-tau, and those with a normal signature had a very less risk to progress to AD, whereas those presenting already preclinically at the prodromal state of mild cognitive impairment with this molecular signature of AD had a high risk to develop AD within the next years, and this corresponds to positive and negative predictive values of roughly 90%. So negative and positive predictive values are completely meaningless without data on prevalence, but the prevalence in the MCI cohort of later Alzheimer's is roughly 20 to 25%. So in view of a prevalence of the hidden prodromal ED of 20-25%, this is highly meaningful in clinical terms. Now, these cerebrospinal driven neurochemical dementia diagnostics of ED has meanwhile been validated by several autopsy controlled studies. Meta-analysis revealed specificity and sensitivity of roughly 85% in more than 20,000 patients worldwide in several independent multi-center studies. And due to this body of evidence, this diagnostic has entered national and international diagnostic guidelines. And meanwhile, research is dedicated to improve the performance regarding coefficients of variation, bias, etc. And there are large EU research initiatives dedicated to this, like Biomark ADP. Now, it's not only useful for the predictive diagnostics, but as you will see here, it can, to some extent, predict the acuity of disease progression. Look at this. So those are patients with a very rapid progression from MCI to AD, those with intermediate speed of progression, and those are slow converters. So there's no correlation of the decrease of 1 to 42. It's decreased, but it's not correlated to the speed of deterioration. Those are the controls. This is completely different for phospho-tau and total tau. Those presenting at baseline with high tau levels are much more at risk for a speedy deterioration. Now, this is another very important landmark paper of Bateman and colleagues published in New York, in New England Journal of Medicine recently, and they investigated those biomarkers, including imaging biomarkers like amyloid PET or glucose PET and structural MRI in a large set of patients with genetic autosomal dominant PRISM1 mutations. And it became very clear that the first changes of biomarkers, look at 1 to 42, it appears 25 years before clinical manifestation of the disease. First, there's a paradoxical increase. It's not paradoxical, actually, but there's an increase. Then 20 years for clinical manifestation, you hit the cutoff line of the population, and when you can diagnose the disease, it's almost at the level of decrease and doesn't change anymore. And if you look at a better deposition in the brain now, which now can be measured by F18 amyloid PET, you already reach saturation when you can diagnose in clinical terms the disease. Now, so this is CSF, but we would like to have blood assays, and I will give 2 examples from our own research. There's another paper and very promising data from the group of Simon Lovestorm from the UK, but due to lack of time, I cannot detail this. So this is a method which some of you might know, at least those working in the field of neurodegenerative diseases. This was developed when I was working at the Max Planck Institute in Göttingen for several years before I started my clinical career. And it relies on the fact that beta peptides change their conformation in the presence of urea due to modulation of intramolecular H bondage. So it's not, in this case, the separation principle is not sieving of the SDS-PAGE gel, but induced conformation changes, which are highly correlated to the primary amino acid sequence and post-translational modifications of beta peptides. And this is really specific for beta peptides. Other groups of peptides do not show this peculiar finding. So first, there's a separation with isoelectric focusing, with immobilized pH gradients, and then we have this urea based second dimension, followed by Western immunoblot with an attomolar detection sensitivity. So this corresponds to several picograms per spot. And we were very surprised that in blood plasma of the patients, the pattern is much more complex than the CSF of the same patients. So this is a 1 to X species, let's say 1 to 37, 1 to 38, 1 to 39, 1 to 40, 1 to 42. So as you can see, you can separate peptides only differing in a single amino acid, which is completely impossible by a 10% T and 5% C separation gel. But to our surprise, there was a reproducible, another lane of beta peptides corresponding to the truncated, aminoterminal truncated species 2 to X, so 2 to 37, 2 to 38, 2 to 40, 2 to 42, missing the aspartate, so we have a shift in the isoelectric point. But at exactly this position, we have a co-migration of the elongated -3 to 40. And I will come back to this because we have quantitative mass spec LC tandem MS data from colleagues from Japan actually pointing to the fact that this is more or less solely -3 to 40. So now if you look at this novel ratio, 1 to 42 divided by the elongated -3 to 40, we were able to correctly classify 80% of patients with early ED, and most importantly, of those preclinical amnestic MCI patients, and those patients were cross-validated by their CSF biomarkers. So we know that unfortunately those patients, due to their CSF biomarker signature, are at high risk to develop AD. And so this is also capable of identifying via blood-based diagnostic those preclinical patients. This is a receiver operate curve characteristics for the standard ratio, the gold standard ratio, and the novel ratio. Now this is a brand new data. So this we patented in 2010, and meanwhile there's this very, I think, strong paper from Japan of a cohort of roughly 60 patients. Every single patient was had been cross-validated by F18 amyloid PET. So in clinical terms, this is a very sound diagnostics. And as you can see, you can nearly distinguish the receiver operate curve characteristics of the F18 amyloid load, but a single scan costs 2,000 euro, and the assay may cost in the end 30 euro. It's almost impossible to distinguish receiver operate curve characteristics of F18 amyloid PET and the novel ratio. And the novel ratio is clearly superior, like in our data, as compared to 1 to 40 in the gold standard ratio 1 to 42 divided by 1 to 40. This is the mass spec data. So this is immunoprecipitation followed by LC tandem MS. You see this is a prototypic pattern in control, and this is a prototypic pattern in an AD patient or preclinical patient with a relative increase of the elongated -3 to 40. Now, here you can see that in contrast to 1 to 40 and the gold standard ratio, the novel ratio is clearly correlating with the F18 amyloid plaque load. Now, in a completely different line of experiments, in experimental models, and human stem cells, neural stem cells. I will not bring those data due to lack of time. So these are data in S555 cells where we were very surprised if we inhibit the beta-secretase base, which is responsible to give rise for this 1-to-X species. As expected, we have a clear decrease of those species, let's say 1 to 40, there's almost a shadow left. But to our surprise, other species were not decreased, but even paradoxically increased. And so this theoretically could be 2 to 40, but now we validated this with mass spec, and actually this is -3 to 40. So there's evidence that those novel species are generated by a novel, so far uncharacterized beta-secretase, which is a very promising novel target. And this may be the reason why the novel ratio depicts a completely different information. It depicts information of the endoproteolytic cleavage balance of 2 different enzyme systems, whereas the gold standard ratio 1 to 42 divided by 1 to 40 is within base. So that's a completely different set of information, and this may explain why it's superior. So I can jump this. So the clinical significance of those aminoterminally modified peptides is that they're highly abundant in human brain. We demonstrated this in JBC before. And that, most importantly, transgenic animal models and human AD brains differ dramatically in the composition of those species. So they are, in relative terms, 10-fold more truncated or modified species in human brain. And they may form a seed for seeded aggregation, so a kind of multiplication procedure or principle as known for prion diseases. Now closing this up, we published in Science 2009 that there is not a single gamma secretase, but there are distinct gamma secretases which give rise to different beta peptide species. So could it be that we have also a distinct beta secretase activity as depicted here? I think our data and data from Eric Portelius and others give clear evidence that this is the case, and those may then be novel therapeutic targets as well as diagnostic targets. Now, the last 2 folds, the last 2 slides, we recently developed a completely new immunosensor relying on Fourier transformed near-infrared spectroscopy as a novel biomarker for prion misfolding diseases. It works the following. On top of a germanium crystal, as part of a flow-through cell, we have antibodies immobilized which catch the antigen, in this case a beta peptide, but it also works for alpha-synuclein, for Parkinson's, for example. And then using Fourier transformed spectroscopy in the near-infrared spectrum, you can have a quantitative readout of the secondary structures. So what percentage is random coil, what percentage is alpha helical, and most importantly, what percentage is beta sheet folded. And our data clearly point to the fact that already at the level of the monomer, patients with AD have a clearly increased ratio of this wrist folding state, which is a prerequisite for following oligomerization in CSF, but most surprisingly also in blood. So these are the CSF data which correspond to a classification accuracy of roughly 90%. Now to sum this up, the molecular pathophysiology of Alzheimer's disease is launched more than 10 years before cognitive brain deficits of Alzheimer's dementia become overt as amnestic MCI. Beta-amyloid-mediated neuropathology with neurotoxic soluble oligomer species precedes tau-mediated neural damage, and moreover, there's a strong evidence for a prion-like disease propagation. A gain of endoproteolytic function in rare autosomal dominant genetic AD is well documented. However, in those multigenetic, 99% of the cases, scientific evidence points much more to a loss of catabolic clearance function. In case of those multigenetic cases, the final molecular cause has not been validated yet, but are beta and tau are at least highly correlated surrogate markers, and we can make use in diagnostics of this fact. Biomarkers can identify preclinical AD, which allows to validate promising preventive treatment strategies in those clinical models. And finally, aminoterminally modified A-beta peptide species are likely to be generated by non-base secretase activity, and they may offer new diagnostic and therapeutic molecular targets. Thank you.